Hello, and thank you for joining us today for BioTek's AppNote Intro live webinar event. The audio for today's webinar will be coming from your computer speakers, so please be sure they're turned on and the volume is up. To ask a question, please use the chat feature. It is located in the bottom left corner of your screen. You can send in your questions at any time. Our speaker will address the questions during the Q&A session at the conclusion of the event. If we do not get to your question, we will do our best to respond by email. Today's webinar on biofuel research will be presented by Paul Held. We'd like to thank him for joining us today, and with that, I'll turn things over to Paul.
Thanks everyone for attending. This is the fourth or fifth of the internal BioTek webinars. It's my second. As Melina said, this webinar is on biofuels. And really what I want to talk about a little bit are some of the application notes that we generated a couple of years back. So biofuels kind of changed since we first visited the research. At the time we were looking at biofuels, gasoline was around $5 a gallon. It's now around $2. The U.S. had only small amounts of oil reserves, and that's turned out not to be the case in terms with fracking. We've got substantial amounts of oil, and the demand was for energy independence, and we're actually an exporter of oil now. So things have markedly changed. There's still a demand to reduce carbon emissions in regards to global warming, and there's still laws on the books that drives ethanol production in regards to gasoline. So there's still some research towards biofuels, but it's been substantially diminished since we first talked about it. Biofuel research can be divided into a multitude of generations. The first generation really is the production of ethanol from corn and sugarcane, and that's really where we're at today. In the U.S., it's corn-driven, whereas down in Brazil, it's mostly sugarcane-driven. And really what we're looking at here is the fermentation of glucose into ethanol typically by yeast. The second generation is really the production of ethanol from sources other than foods, such as corn. It's really from woody biomasses, and it's digestion of cellulose rather than starches. And last but not least, the third generation is really the production of biodiesel from algae, where algae will actually produce lipids under certain conditions. So I think what we're looking at in long term is the research in regards to algae, but we'll talk about the first two generations as well. So ethanol production, it's been around for centuries. Some people say this is the formation of civilization was the result of the ability to ferment grains. It's typically done with Saccharomyces cerevisiae. And it's kind of a simple reaction where we convert one molecule of glucose into two molecules of ethanol. So the generation of glucose is really what drives the generation of ethanol. Starch in grains is the primary source of glucose for the fermentation. Starch is water soluble, and it's basically just a glucose polymer. There's two types of bonds. There's an alpha-1,4, which is the long linear chains. There's an alpha-1,6, which is basically our branch points. So the research around starch digestion really is the research around being able to digest these two bonds into simple glucose. There's a number of different substrates. One of the most useful ones in terms of microplates is actually a starch compound that has multitudes of fluorescein moieties on it, to the point where, as it stands, it's self-quenching, and only when it's digested by amylase, which is the typical enzyme for digestion of starch, that the fluorescent molecules are liberated, and you see fluorescence. You can see in the graph to the right, there's a number of different sources of amylase, and they've got slightly different activities in regards to their stem concentrations. Typically, they're either from bacteria or fungi. Actually, we've got an example of both here in this example. But nevertheless, really what we're looking at here is the digestion of the non-fluorescent substrate into a fluorescent compound in vitro in microplates. With the advent of microplates, we can certainly do a certain amount of optimization. Really what the goal here is to look for enzymes that are both temperature as well as pH resistant. The example on the left is a temperature profile where you're looking at, I think this is the aspergillus reaction, where it's actually quite resistant to temperatures up to even 70 degrees Celsius. But on the other hand, it's not necessarily that pH resistant. So it's really a combination of both temperature as well as pH. And ideally what you want is high temperature and low pH. Those are reaction conditions that favor glucose production. Second generation, as I said earlier, it's actually from woody stocks such as grasses and wood chips. Primary constituent of grass is cellulose, which is also a glucose polymer, but it's actually a beta 1,4 bond. So it's actually the opposite bond as the starch. But there's other components in woods and grasses such as hemicellulose and lignin that actually have some utility in terms of fermentation as well as other products. This kind of schematic, what we're looking at is the different uses of the plant biomass. It can be, as I said, it can be broken down into cellulose, hemicellulose, and lignin, two of which are useful for ethanol production, the cellulose and xylose, whereas lignin is usually done for digested into specific chemical solvents. In addition to ethanol, there's a number of other different products that can be made from these plant sources as well. And I think with the loss of the diminished drive for ethanol production as a result of gas prices, these other uses will actually take a lion's share of the new research. In regards to glucose generation, as I said, it's a beta 1,4, so it's actually the opposite of starch. But cellulose is basically a long linear chain, so we really have one bond. So if there's an enzyme that can break cellulose down, you can really produce large amounts of glucose. And then once the glucose is produced, then you can then ferment it to ethanol, just like you would a starch-based source. Most of the compounds most of the enzymes that perform this are actually isolated from fungi. Aspergillus niger is probably the most common example. It's a very common fungus. It's actually the one that forms black spots on vinyl-sided houses as a result of mulch being below them. But it's a really good source of digestive enzymes, and it's the primary digester of fallen tree trunks in forests. So, in essence, what we're looking at here in terms of enzymology is the production of glucose. So, in regards to the assay, what we're really going to do is digest cellulose into glucose, and by doing so, what we're going to measure the end product, which is glucose. And it's a coupled fluorescent reaction where we produce resin-rupin as a result of glucose oxidase activity on glucose. So the more glucose present, the more glucose oxidase activity there is, the more fluorescence produced. So just an example of the titration curve we would generate. From there, we would use that titration curve to calculate the production of glucose from cellulose activity on non-fluorescent cellulose. Really what we do is we digest the cellulose with enzyme, specific different enzymes, and take a small aliquot and assay that for glucose production. Not surprisingly, the more enzyme we have, the more glucose is produced and more fluorescence. As you can see, it's actually a concentration-dependent reaction based on the amount of enzyme you have. There's also a number of cellulose substrates that are fluorescent. The compounds are proprietary in terms of their structure, but in essence what we're looking at is the amount of fluorescence produced is proportional to the enzyme concentration. In this graph, the greater amounts of enzyme present, the more fluorescence is generated with these types of assays. Again, you can do an enzyme titration. So if you just titrate the cellulase activity, it's proportional to the amount of fluorescence produced. These kind of assays, again, can be used to titrate pH, whereas in the case of Aspergillus, it's actually quite resistant to low pH, which is really what you want. Acidic pH is good at digesting plant components. So if you have an enzyme that can actually then break down the cellulose at low pH, that's an advantage as well. Xylane, as I said, is one of the other components of wood materials. It's a slightly different sugar molecule as well. It's actually a component of hemicellulose. The idea here is they also want to digest this pentose or 5-carbon sugar as well to produce ethanol as well. In fact, xylose is the second most abundant sugar in plants behind glucose. Just an example of some of the research that other folks have done where they really engineered bacteria in yeast to utilize these non-glucose components in terms of the production of ethanol. The idea being that they can digest hemicellulose and xylose to produce ethanol as well as glucose in parallel. So the idea being that they can produce more ethanol with more substrates. There's a number of different fungi that, like I said, can digest these things. There's also fluorescent substrates that measures xylase activity, one of which is basically just a methylene derivative. The idea being with enzyme activity, you're going to liberate this fluorescent substrate. The original substrate is non-fluorescent and the product is fluorescent. Again, the idea being that the more fluorescence you have, there is more enzyme activity. And these are just examples of two different experiments with thermomyces. As you can see, with increasing enzyme concentration on the left, you get more activity. And this Thermomyces fungi is quite temperature resistant. As you can see, even up to 65 degrees C, it maintains full activity. If you look at pH, so it does have a pH profile that's slightly different. It actually works quite well around neutral pH, 6 to 7, and then rapidly drops off as the pH is either too low or too high. So again, these are all kind of classic enzymology reactions that are often performed with different enzymes. And really what we're looking at here is the production of mutant enzymes that show more activity under desired conditions such as low pH. And in the end result is the production of ethanol. It's really what we wanted. The last piece of the puzzle in terms of the second generation activity is the ability to quantitate ethanol production itself. You can certainly do it at different scales. Densitometry is what's normally done in kind of the home brewing in the commercial beer industry, whereas in the organic systems, a biochemical assay is done. Again, it's a coupled assay where we've got alcohol oxygenase coupled with a non-fluorescent compound that produces resirufin. Again, the more ethanol that's present, the more alcohol oxidase activity there is, and the more fluorescence is generated. So you can see in the calibration curve to the right, you can detect as little as 0.003% ethanol. So very minute quantities of ethanol can be measured, but on the other hand, also quite high concentrations of ethanol, approaching 100%, can be measured with these assays. This is just an experiment we ironically did in the lab where we actually had fermented beer in the lab for a while. As you can see over time, the ethanol increases in fermented beer with fermentation, reaching a saturation point. And we actually did some field testing as well with a number of different commercial breweries where we looked at what the reported concentrations of ethanol versus what we measured. And for the most part, the assay correlates quite well. Some of the beers that have high sugar content tend to read a little high, but most traditional beers are quite accurate using kind of biochemical reaction. Third generation, it's not ethanol based, it's actually diesel based or lipid based, and it's based on the use of algae. Algae are single-celled organisms. Most of them are photosynthetic. In other words, they have chlorophyll and they can utilize energy from the sun. The idea is they can produce both biodiesel or lipids as well as a biomass that can be used other means such as burning. Cell growth quantitation is actually quite simple. In essence, grow your yeast, not yeast, your algae cells in flasks. You take a portion of that flask and put it in a well of a microplate, and then measure its absorbance. You can see this these two graphs. One is absorbance, and it's really not true absorbance. It's really light scatter. You can see a certain profile. There's some kind of peaks to the left. That's really the result of chlorophyll absorbance. There's actually a fluorescence component to chlorophyll, so you can actually measure the fluorescence of the chlorophyll in cells as well. In regards to light scatter, really what you want to do is avoid areas where there's absorbance, and you really want to look at just light scatter. So we typically use the 600 nanometer range. It's actually the region of the absorbance spectrum that's quite low. As I said, it's a light scatter assay. The premise of it is that cell suspensions will scatter light. It's not a true absorbance assay as defined by Beer's law. It's actually diffracted light, and the concept is that the diffracted light caused by the suspension will basically bend the light such that it doesn't doesn't reach the detector. So any light that doesn't reach the detector in our readers is reported as absorbance. What we found is that the absorbance is proportional to cell number, but it's also a function of cell size as well as optics. So different readers will have different results from the same number of cells, and different cell types with different size will have different results as well. So depending on what cell type you have, you really need to have a calibration curve based on each cell type. And this is just an example of that, where we have 3 different algae strains that we did serial dilution on and looked at their absorbance. And as you can see, they're all linear in terms of their dilution, but they're all different from one another. And this is based on differences in cell size. Likewise, fluorescence, you can use fluorescence to measure cell number by diluting cells in this same fashion, and you measure the fluorescence, you see a linear response as well. So we compared absorbance and fluorescence with kind of the classic algae, Chlorella vulgaris, and as you can see, both absorbance and fluorescence match up quite well. Really what this indicates is that the average amount of fluorescence in each cell is the same, so the more cells you have or the more absorbance you have, the more fluorescence you're going to have. Likewise, we plotted cell growth over time using where we monitored either fluorescence or cell number. And as you can see, over time, the cultures will grow and rapidly increase in fluorescence and or absorbance. And at some point, they reach a saturation point where things level off. So the real gist of the matter is the ability to produce lipids. So the first thing we want to do is look at rich versus minimal media. The idea being that rich media allow for cell growth, and we know that minimal media actually provides some means for lipid production. So the graphs on the left really show the change in the absorbance, or basically the change in cell number over time with different rich medias. TP and TAP are considered very rich media, whereas BG11 is a minimal media. As you can see, the rich medias are very good at growing cell numbers. Likewise, if you take a rich media and then subtract a component such as nitrogen or sulfur, you see substantially less growth in those medias as well. So complete media that's rich produces large cell numbers, and anytime you have something that's lacking, cell growth drops off quite rapidly. As I said, the game is to produce lipids. Lipid production in algae is a stress response. Normally, rapidly growing algae cells have very little lipid, but when they have some sort of they meet some sort of stress, whether it's lack of nutrients, they basically go into a starvation mode where they start producing lipids. The advantage of this is we can actually track these lipid production using a stain called Nile red. It's very non-fluorescent in aqueous environment, but fortunately it partitions into neutral lipids such as triacylglycerols, and it's quite fluorescent when it's in a lipid environment. As I said, it's a stress response, so there's a number of different things that can cause the formation of lipids in algae. The one we've talked about the most is nutrient deprivation, but also if cells perceive a change in pH or a change in salt levels or any kind of physical stress, they'll often switch from carbohydrate-based metabolism to the formation of lipids. The assay is pretty easy. You pipette cells into a well. You measure their absorbance as you would to quantitate cell number. Then you add some Nile red and they incubate for a short period of time and measure the fluorescence. If you look at the loading kinetics of Nile red, it takes place within 3 or 4 minutes, and it's quite stable for a number of hours. Not surprisingly, if you have cells that have produced lipids, increasing cell numbers will cause an increase in fluorescence, and it's a result of literally cell fluorescence, as you can see. If you don't put Nile red in the reaction, you get very low fluorescence. So the effective deficiency of nutrition is really what we're looking at here. So in regards to the production of cell number, as we said before, complete media produces large numbers of cells, and anytime you remove one of the components, such as nitrogen or sulfur, the cell number over time diminishes. On the other hand, those reactions, those conditions that have low cell number actually cause the cells to produce large amounts of lipids. In fact, the nitrogen deprivation actually causes the most production of fluorescence and or lipids as compared to the complete media. If you look at the full change, you can see both the nitrogen deprivation and the sulfur deprivation are much better at producing lipids in individual cells as compared to the complete media. On the other hand, the complete media has large numbers of cells. So the idea is you produce large numbers of cells in complete media first. To obtain a significant number of cells, you then switch to some sort of deprived media, and that large number of cells then rapidly produces. And if you have cells that have large amounts of lipids, and then you basically return them to a complete media, they quickly digress and use up the lipids quite rapidly. And as you can see, an example of the complete curve, really what you're looking at here, you see a small increase in signal. That's actually the result of the physical stress of centrifuging the cells to switch medias. So in conclusion, really what we're looking at in terms of biofuels for the most part is the production of glucose for fermentation, whether it's from a food source such as corn or a woody plant source such as grasses and woodstock. Regardless, there's a fairly complex enzymology. This kind of enzymology can be done in microplates quite well. And these are, not surprising on readers, I think provide a great means to optimize a number of different parameters in terms of reactions on a very small scale, with the idea being that once these parameters are optimized on a small scale, that information can then be used to scale up to much larger scales kind of the industrial scale. There's a number of application notes out there on the website. Sure that if you want more complete information, please visit BioTek's website. And if not, you can always contact me. Thank you very much.
Thank you very much for joining us today. Recording will be uploaded to BT Resource along with a copy of PowerPoint slides. You can locate the zip files that contain the recording and the PPT slides under Document Search, Document Type equals Webinar Search. Again, thank you very much for joining us, and if you have further questions, please feel free to reach out to Paul. Thank you.
Thank you.